Hey guys, welcome back to our third set of notes here about plant tissues. We're going to go ahead and move to our next plant organ, leaves. And you guys probably know this, of course, but leaves primarily perform photosynthesis, where they take carbon dioxide gas and sunlight, turn it into sugars. Very important for plants, very important for ecosystems. And then gas exchange, of course, they're going to let oxygen gas out of these little pores called stomata, and then let carbon dioxide gas in through the same little pores called stomata. Leaves vary in size, shape, and surface specializations, of course. Every leaf is different, and their internal structures vary a little bit as well. So, there are several different ways to describe leaves. These are just a few. Elliptic is this kind of oval sort of shape. If they're a palmate leaf, there are leaflets that radiate out like fingers from a palm. Lobed or lobate has these sorts of lobes in one leaf. This is pinnatisect, which has little leaflets coming out, kind of like pinnate venation. It has pinnate venation, but also has leaflets that are organized sort of pinnately. This is odd pinnate, where instead of having it come off of the stem in several places, it just comes off at one end. And there's a few other names here too: lobed odd pinnate or bipinnate, and elliptic odd or pinnate, and you're not going to really need to know those, I don't think, for a test. But there are several ways to describe the way a leaf looks, and from the leaf itself, you can just about always tell what plant it's from. So there are some specialized leaves. Not all leaves just lay flat like these and collect sunlight. There are leaves that perform special jobs. For example, this is the pitcher plant, and it has a leaf that's modified to be this big container that traps water. It even has a hood that comes over the top, and bugs can fall in there and die. Their plant looks like this, called Jack in the Pulpit too. That might very well be what this one is. And then it digests those bugs in here. This is a sundew leaf. It's covered in little sticky pads, and as a bug gets on the leaf and is kind of wiggling around, the pads all stick to the bug. The leaf curls in, and it digests and eats the bugs. There are also bracts, or floral leaves. The Christmas poinsettia that we buy does not have red flowers. It has red bracts. They are leaves that are brightly colored to attract pollinators to the very small, inconspicuous flower. So when you look at leaves, there's a couple different leaves. This leaf has a petiole, which is essentially a small stem that holds a leaf out away from the plant. Not all leaves have petioles. Some leaves are what's called sessile. This is a sessile leaf from a blade of grass, where it doesn't have a petiole. Rather, the leaf wraps around the stem of the grass plant with a sheath, and that does still come out at a node, just like this leaf comes out at a node. This is the leaf blade in both cases. One's parallel venation, so you know it's probably what? A monocot or a dicot? Probably a monocot. This one has netted venation, so we know it's probably a dicot, but it still has a blade, and this still has a blade. Still have a stem, we still have a stem. This one has a lateral bud. This will become next year's branch. It'll grow out after this leaf dies and fall off. This will grow out next year into another branch or twig. Whereas you don't, of course, see that in grasses because they're going to survive for 1 year, and that's going to be about it for most of them. So those are some different designs in how you can put a leaf on a stem. So if we look at just the leaf itself, there are different aspects of the leaf if you cut it in cross-section and look at it under a microscope. The mesophyll is the middle of the leaf between the top and bottom layers, the epidermis, the upper epidermis, the lower epidermis. Between that is the mesophyll, which is the middle layer. And the photosynthetic parenchyma is in here with air spaces, so a lot of the photosynthesis is happening here. It's the bulk of the leaf by far. There are leaf veins, and leaf veins are the special name we give to vascular bundles, which is xylem and phloem, that are running through the leaf. The epidermis, not surprisingly, is the outermost cell layer of a leaf. It's the one cell layer thick, with very, very thin epidermis. It can form all sorts of unique outgrowths like hairs, scales, spikes, prickles, things like that. And oftentimes it will secrete a waxy cuticle that slows water loss and makes the leaf survive just a little bit better in a drier habitat. And so here you have a leaf in cross section. This is the vein, xylem and phloem inside of the vein. More than likely xylem here is these very large vessels, phloem here where the word vein is. An upper epidermis, a waxy cuticle, a lower epidermis, and a waxy cuticle. Right here, you know it's the lower epidermis because these two fellows here, that guy is one guard cell, that guy is one guard cell. And the little hole that they would have open is called a stomata, which you already know. And so you know that's the lower surface. Stomata are generally, I almost want to say always, stomata are always located on the lower end of the leaf because that's the way we can get gas in without losing too much water through evaporation. It's really hot on the top of the leaf, so it's easy to lose a lot of water. And then all this stuff in here is mesophyll, middle cell layers. And of course you can see the cells with the chloroplasts in the cells and some of the white air spaces that are trapped within that leaf as well. So that's your leaf. That was a monocot, of course. This is a dicot. So dicot, we're going to get a few more leaf veins. Still a similar design, except for we're going to split our mesophyll up into a palisade mesophyll, where the cells look long and column-like, and then a spongy mesophyll where they're kind of random and I guess circular, more or less. So there's two layers of mesophyll, palisade on top, spongy on the bottom of the leaf, but you still have the same design, upper epidermis, cuticle, lower epidermis, guard cells, stomata, that stuff's all in this dicot leaf as well. So here's a little better picture of the palisade mesophyll and how they're long and tall like a column, and then the spongy mesophyll here, which are all these sorts of odd shapes with abundant air spaces in the spongy mesophyll. We pack the upper layer a little more tightly with cells because that's going to do the majority of the heavy lifting when it comes to photosynthesis. So it makes sense to have a lot of cells, a lot of surface area towards the top where you're going to be doing that photosynthesis. Then we have xylem and phloem traveling through a vascular bundle, and that's how a leaf is designed. So that's leaves. Let's go to roots. You guys know what roots do. Roots grow down into the soil. They take up water, they take up minerals, and they keep the plant from tipping over. Right? Anchoring the plant to the soil is very important. Sometimes they can be used for storage, and we can take those storage roots out of the ground and eat them. Most certainly a carrot is probably our most common example of that, but there are other carbohydrate forms in roots that we can eat. Dandelion roots, for example, you can eat those as well. Just make sure they weren't sprayed with anything weird first. So there's a couple different root systems. If you are a monocot, like a grass, you have a fibrous root system where just little fibers grow down randomly, and we've talked about that already. There are adventitious roots, which can grow up from the stem, like a prop root in corn that props it up against the wind. There are lateral roots, which grow out to the side in very shallow soil to keep the plant stable and to take up additional nutrition. And the vascular cylinder is what's going to divide up our cortex and our pith, kind of like what we've seen before. So in a dicot, you're going to see less of a fibrous system and more of a taproot system. So here you have this little marigold. It has one primary taproot coming down with little branching lateral roots going out to the sides. Then here's the carrot, one primary taproot, which is what we're generally buying in the store, with a variety of small little lateral roots that we are cleaning off and taking off before we eat the carrot, right? So that's the taproot system in a dicot, and they have a central vascular cylinder runs right up the middle of the taproot. So the vascular cylinder in roots are called steles, and there's all sorts of different types of steles, but we won't get into that necessarily. Just know that that's what you call your vascular bundle in a root. It's a column of vascular tissue, xylem and phloem. It runs lengthwise through the center of the root. And we have a few different types of tissue layers. Really, I don't know if we would call them tissue layers, cell layers in a root that we want to know. The epidermis is on the outside, like we've seen before. And we also have a circular layer inside the root called the endodermis. Endo means within, derm means skin, so this is the layer of cells within the skin. It's the outer boundary of the vascular cylinder. That's important to know, and it helps control which solids are taken into the plant's vascular system. It's like the guards before you get to xylem and phloem. We're picky about what we let into our root because we certainly don't want to let some sort of parasite or little bug get into our xylem or phloem, or they could travel throughout the entire plant and infect the whole plant, and that would be bad. So endodermis is making sure that's not happening. And then the pericycle is just inside of the endodermis, and it's just one little cell layer thick, and its job is to grow lateral roots and root hairs in a young root. So that's a pericycle's additional growth there. And so here's kind of what it looks like. Monocot root anatomy, one of those fibrous roots. You get a ton of this stuff is really just cortex, that ground tissue. All of this from right here to right here, it's all just ground tissue. This is the epidermis all the way out here. and a variety of little cells. There may or may not be mycorrhizal fungi growing in with this root, helping it take up nutrition, because they will oftentimes form mutualistic relationships with fungi in the soil, which is kind of cool. And so this little tiny baby right here in the middle is where all the business goes on for transporting sugars, water, ions, vitamins, minerals, things that it needs. And so here you have the endodermis, just this one little layer of red cells. Inside of that, you have the pericycle, one little layer of, I guess, clear blue cells. And then you have a pith, which is very small in this example. And then you have xylem, which is these big white vessels. And then the blue stuff is phloem. The phloem's on the outside. And so that's how the vascular bundle is arranged in a monocot root. Now, a dicot root's similar. It tends to look more like a cross or an X as the way it's designed. This one's a little crazier looking, but this you have the epidermis on the outside again with a big old cortex and ground tissue throughout the middle, and then way in the middle of the root you have the same thing, endodermis just like before, pericycle just like before, phloem on the outside just like before, xylem on the inside makes more or less, it should make a little X or a cross. That's the way I learned it. This picture is not a perfect one, but it should kind of make kind of a line right there. And then you should have phloem just filling in the area around it. And so that's the dicot root anatomy, very similar to monocot. So primary growth is just a plant growing longer. A taller plant or a plant with a root that grows deeper into the soil, that's primary growth. The production of leaves, that's going to happen right after primary growth. We try not to get branches that are exceeding the height of the plant, and so that's called apical dominance, where the apex of the plant, or the top of the tree, gets more growth hormone than everybody else. And they don't get growth hormones until the top of the plant is quite a bit ahead of them, and then we start to produce leaves. They originates in the apical meristems, like we mentioned before. There's the ram and the Sam. The root apical meristem makes roots longer, and the shoot apical meristem makes the stem taller, and that is undifferentiated cells that are pretty much constantly dividing. It's always dividing cells in the plant. That's called a meristem. And so here we have, if we take the tip of a plant that's just starting to grow, this is the shoot, and we take a look at what's going on here. We have the apical meristem, which we mentioned was the actively dividing cells. That's right here. If you were looking for mitosis, phases of mitosis, this would be the place to look. You have little immature leaves right here that grow beside it. We can call those leaf primordia, or the first leaves, and they're going to start to grow out as the apical meristem continues to grow out in this direction. Then you get several layers. You get a protoderm, which will become the epidermis, and you get a procambium, which will become xylem and phloem. So those are kind of nice. Protoderm before the skin becomes the epidermis. Procambium becomes xylem and phloem. The ground meristem is going to become ground tissue. This is growing out little leaf hairs. There's axillary bud meristems. And so we regulate how much a plant grows with a hormone called auxin. And so once this guy gets tall enough up, they'll get a little more auxin and they'll grow lateral buds out. Here the procambium started to differentiate a little bit into true xylem and phloem, what will eventually become a leaf vein. And then all this stuff in here is just ground tissue like we've talked about before. It takes up space. And so the cells are dividing at the top. They're differentiating towards the bottom where they're becoming true xylem and phloem. And then once they're the type of cell they need to be, then they just enlarge or grow longer, but they don't actually divide any new cells. The only new cell division occurs at the very end of the shoot and the very tip of the root, and then everything on from there is just cells becoming longer and becoming differentiated into the jobs they're supposed to do. So that's kind of how plants grow. Now here's the same picture in the root. You have the apical meristem, which is right here. You've probably seen this in the onion root mitosis lab. This is where the cells are actually dividing. This is the root cap, so this kind of protective layer that makes sure that this guy doesn't get damaged, because if he gets damaged, we're in a ton of trouble. There's ground meristem, which is just going to become ground tissue. There's procambium, which is going to become xylem and phloem, which is right here in the middle. And then there's protoderm, which is going to become the epidermis as the plant continues to grow. And so it's really the same thing, division at the bottom. Cells divide here. That's the only place you're really actively dividing. You can actually see it. That looks like telophase right there maybe. You have differentiation, where they start to become specific types of tissues, and then they don't divide anymore up here. They just grow longer, and that's how the root goes longer. Okay, so secondary growth is growing out wider. For a tree, they not only get tall, but they also grow out wide. For grasses, you don't see this a lot necessarily. You get a little bit, but really we see it in dicots, especially trees. And that secondary growth is what is responsible for laying down layers of wood year after year. And there are two types of lateral meristems that are responsible for this, and that's the vascular cambium and the cork cambium. And they do two very different jobs, but I'll kind of show you where they're situated here. So this is the cork cambium. The cork cambium is producing cells out this way that are essentially becoming bark. They're becoming cork cells, which are tough, dead cells, and then they end up becoming bark. Now, it's also producing tissues in this vascular cambium. The vascular cambium is the orange line, and that's growing tissues in and out. It grows phloem out this direction, and it grows xylem in this direction. And so as the phloem gets pushed out, older cork cells get pushed out further, and they dry out and they die and they become bark. And as xylem gets pushed in, it makes what we call wood, and it makes annual rings. Now xylem's being made all the time, and so it's very useful to us to know that because it tells us a lot about our climate. So here's kind of what your tree might look like. The outer part of the tree is called sapwood, which is recent wood. So 1, 2, 3, 4, 5. This is about 6 years ago is what this picture is showing us. Heartwood is the older wood that's so far from the vascular cambium, it's not getting a lot of water, and it'll accumulate a bunch of resins, and it can often die. And an old tree can frequently die of what's called heart rot, and that old stuff will die and rot out. And then the layer between the wood and the bark, this little orange sheet, that's called the vascular cambium, and that's really the business end. Pushes xylem to the inside, phloem to the outside, and the vascular cambium is the reason this tree is growing. Then there's secondary phloem, which is almost bark, but not quite. And some people and some tribes have actually eaten that before because phloem is a sugar source, and if you take the bark off a tree, there's an inner bark, this white fibrous sticky stuff, and in most cases, you can actually eat that if you're really, really hungry. And then the stuff that gets pushed way out here is the cork cambium and the cork, and a little bit of phloem, and that's what dries out and becomes bark. So if you want to kill a tree, it's not that hard. You don't have to cut the whole tree down. You just scrape the bark off all the way around. It's called ringing it. Ring the tree, scrape all that bark off, and you get that vascular cambium off, which is the white fibrous stuff inside of the phloem, and that tree will die. It can't grow anymore. It'll be dead. It'll be dead next year. You come back, cut it down, it'll be all dry. You can use it as firewood. So that's kind of how trees grow, and we can count these rings back because in the springtime they get a lot of rain and they grow really big xylem vessels, and so they're like lighter colored. And in the winter and the fall they get very little rain, and so they still try to build xylem. There's just not much water to pump, so they build small xylem vessels, and those small xylem vessels show up as a dark band separating springs. And so you can kind of get a pretty good count on how old a tree is. Just spring, winter, spring, winter, spring, winter. Just count your way back. Cool. So here's the dicot stem we've cut in half with pith on the inside. We have an endodermis. We have our xylem, primary and secondary, which is just xylem getting pushed to the middle. And that's what comprises wood, really, is that secondary xylem. Here we have a little vascular cambium, pushes phloem to the outside. We have a cork cambium that makes cork, and as that gets pushed to the outside, that will eventually become bark. And all the cork and secondary phloem together are called a periderm, or around the epidermis. Cool? So that's basically how a tree is designed. And so if we count back with tree rings, we can get the seasonal growth I was explaining to you about. There's early wood and late wood, or spring wood and summer wood. And early wood is large diameter vessel elements and really thin vessel elements. And late in the year, they make these small little tiny vessel elements with thicker walls in them. And each band tells us 1 year. So you can cut down a tree and say, okay, if this is right now, then that's 1 year. Last winter, 1 year. And then it was born then, so that was year 1 of its life. So it's a 3-year-old tree, 2.5-year-old tree. So you can get a pretty decent count on these things. And here's a really cool picture. This is direction of growth: early, late, early xylem, late xylem. So spring, fall, and winter, spring, fall, and winter. So you just count it back. Born, 1 year old, 2 year old, 3 years old, 4 years old, 5 years old, and I don't know how long it goes on, but right there we're at a 5-year-old tree. And so it's kind of cool because you can get tree rings that date back quite a long ways, depending on how you're able to get these trees cut down and how old these trees are. And they can tell us a little bit about why some civilizations may or may not have survived where they tried to, or why some survived but just barely. We can look at things like when settlers made it to America, when they were trying to survive, they had just a heck of a time, and half the population, 3/4 of the population was dying. And when we look at tree rings from trees that were alive when people were trying to settle America, well, settling, they succeeded. We're here. We see that they were in an awful drought. The drought lasted like 7 years, and so it gives us ideas about why societies struggled and when they struggled, and what our climate was like, 100 years ago, 200 years ago. As long as the tree is old, we can kind of track our way back. And these really thick rings right here, that's a lot of rain. That's a good year. And you get to some of these really tiny bands, and those are like right here. That's not good, not good, not good, not good. So you get 6 years of just bad weather, and then maybe a fire. And then next year, good, 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 bad. And so it gives you kind of an idea of what the climate might have been like. And so here's your 1587 to 1589. Pretty serious drought. Those are small little lines. And you get some couple of really good years here with lots of rain. Another kind of not great time, bit of drought. Really, really good time. And then a really long drought. Like, look how small these lines are. It's 1, 2, 3, 4, 5, 6, 7, 8 years or so. 8 years of really, really, really dry years. And so it tells us a lot. So there's your points to ponder. That's a chapter. Thanks for your time.